Set to go? I'm too excited today. You don't have to necessarily do it today, but you do need a broad culture of the unknown for a lab on Wednesday. If you have one that grows a little bit slow, you might want to do it today, otherwise you can do it tomorrow. And then don't forget to just take your plates out of the incubator and put them in the refrigerator. I just checked a few of them, and I'm seeing a few different things pop up on those plates that we didn't see before. One of the things to be careful about on that is that if you do see, especially a single isolated colony someplace out there that you haven't seen before, look at the bottom of your plate and see if it's on one of the streak lines that you made on the plate. If it's not on one of the streak lines, then very likely it's a contaminant. Okay, so be aware of that. But they probably shouldn't be in there any longer, so treat it up. Questions, anybody? Okay. Well, last time we were talking about the fact that in the microbial world, metabolically, things are a little bit more complicated than they are in the eukaryotic world. And specifically, we talked about how you can begin to deal with the diversity of metabolic strategies in the microbial world by characterizing organisms based upon two things. Number one, where are they getting their energy, and where are they getting their carbon? In the past courses, like cell biology, we've simplified it because we aren't dealing mostly with microorganisms. And we basically dealt with these two particular categories up here, the photoautotrophs and the chemoheterotrophs. And usually we have just abbreviated that by calling organisms autotrophs or heterotrophs. But in the microbial world, you've got to be a little bit more precise than that, because there are some other strategies for metabolism besides those two. In reality, there are really four strategies represented in the microbial world. We're going to spend most of the time talking about chemoheterotrophy, because that's where the majority of organisms that we're going to work with, that's the strategy they're going to use for their metabolism. And that would include all of the known organisms that we use in lab as controls, and also all of your unknowns as well. However, we will talk a little bit about chemoautotrophs as well, because they're pretty interesting organisms, and it's a very different adaptation to surviving in the world. And then we'll spend just a little bit of time talking about the two strategies of phototrophy, the photoautotrophs and the photoheterotrophs. So let's dig into chemoheterotrophy just a little bit. Chemoheterotrophs have one of three different strategies for their metabolism. They can either carry out aerobic respiration, anaerobic respiration, or fermentation. Some organisms can do all three depending upon their conditions. Is oxygen present? Is it not present? Some organisms are restricted in the terms of the types of strategy they can utilize. Some will only ferment. Some will do both aerobic respiration and fermentation, but not anaerobic. And so there's a lot of different possibilities depending upon the circumstances. And as far as your unknown is concerned, one of the things we really do need to nail down is this distinction between whether your organism is strictly or obligately aerobic, or whether it has the capability to do aerobic and anaerobic and or fermentation, meaning it would be facultative. So that's something you're going to do on Wednesday when we inoculate unknown and put it in the gas bag, the candle jar. And then later on, in a couple of weeks, we're going to spend a fair amount of time kind of determining your organism's abilities to ferment various substances. So can they use a food? And if they ferment it, what do they produce as byproducts? And again, that will be useful information about your unknown, particularly if you have one of those pesky little gram-negative rods that is very difficult to see anything with, particularly regarding structural information. So here's our basic overview. This is just kind of a review slide of aerobic respiration, anaerobic respiration, and fermentation. And again, one of the important things to remember is that anaerobic respiration and fermentation are not the same thing. And that's a common misconception that a lot of people have, and oftentimes feel that those two terms can be used synonymously. They can't. Okay? Anytime you see the word respiration, that implies the existence of a Krebs cycle and an electron transport chain. Obviously, in fermentation, which isn't a type of respiration, there is not a Krebs cycle and an electron transport chain. Different end products are produced as a result of these particular pathways. A lot of similarities in the pathways of aerobic and anaerobic respiration. Really, the major thing is what is being reduced in the electron transport chain. Where are those electrons that are being removed from your food molecules ultimately ending up? Are they being grabbed by oxygen and reduced to water? Or in the case of anaerobic respiration, are they being grabbed by some inorganic compound that has oxygen in its structure, but is not molecular oxygen? Okay. We did talk about the fact that there are some differences in energy yield, aerobic respiration being more efficient than anaerobic respiration. And that manifests itself in the fact that organisms that are growing aerobically will have better growth than those that are growing anaerobically. Okay? So if you've got a facultative organism, if you grow it in a regular incubator, in the same period of time, you'll get more robust growth on a plate than if it's growing in that gas pack where it's deprived of oxygen and therefore has to rely on this particular pathway. One of the things that we're going to encounter in the aerobic respiration, and specifically looking at the electron transport chain, is differentiating between some of the types of compounds that make up that electron transport chain. Remember that some of these compounds that make up the electron transport chain are called cytochromes, and these are molecules that contain iron. And like hemoglobin, these molecules can be alternately oxidized and reduced, so they can gain electrons, accept electrons. One of the things we'll be interested in some case is what is the final compound in that electron transport chain? What's actually donating the electrons to oxygen to make water? Another way to say that is what is the terminal electron acceptor? In some organisms, but not all, that final compound that donates the electrons to oxygen in the electron transport chain is a compound called cytochrome A3. But that's not the case for all organisms, okay? So this is a kind of a diagnostic trait you could use. There is a test that we're going to do later in lab that's going to be pretty important for some of you in terms of where you go in the key. And that particular test is called the oxidase test. And a positive oxidase test indicates the cytochrome A3 as being that terminal electron acceptor. If you get a negative test, they're using something else. Okay? Their electron transport chain is constructed a little bit differently. And this is a really easy test to do. You just take a strip of material impregnated with some chemical and smear your organism on there. And if that chemical changes color to a blue-purple color, that's indicative of a positive oxidase test. So we'll be doing that test later on. Now, with fermentation, it gets a little bit more interesting because we don't have an electron transport chain. Remember that the purpose of the electron transport chain, the real purpose of it is to recycle your reduced NAD and FAD. You need to get those back to the oxidized state. The cool thing about the electron transport chain, however, is while that recycling is taking place, hydrogen ions are being pumped across the membrane. And because hydrogen ions are accumulating on one side of a membrane, if they're allowed to flow through a channel protein, that ATPase channel protein in the membrane, then they can produce energy by oxidative phosphorylation. So that's where we get much of the energy payoff in cellular respiration, is because of this electron transport chain, because of the fact that those organisms are pumping hydrogen ions, and then using that hydrogen ion gradient to make ATP. Well, in fermentation, we still have to recycle the reduced NAD that's produced in glycolysis. But we do so in a different manner because there's not this electron transport system to do it. So we have to use the reduced NAD to provide electrons to then convert pyruvate into some other final fermentated compound. And that can be ethanol, it can be lactic acid. I'll show you a number of examples of different types of fermentated byproducts in just a second. But there's a whole variety of different things that can be produced as a result of fermentation. So we'll get into that a little bit as well. Okay, where are we going next here? Okay, so again, this is just kind of some review material talking about how in respiration we have glycolysis, we have a Krebs cycle, and they're not showing up, they have an electron transport system. In fermentation, we don't have those Krebs cycle and electron transport chains. Remember also, this particular slide shows us a little bit, is that there are two roles to respiration and fermentation both. And that is providing energy in the form of ATP, but also carbon skeletons can be generated that can be used in biosynthesis. And so pyruvate could be broken down into CO2 and water to make the maximum amount of ATP, but pyruvate might be shut off into other metabolic pathways to make amino acids, fat metabolites, etc. Things you need to build new cells and to sustain growth. Don't just think about cellular respiration and fermentation as just means to produce ATP. There's that biosynthetic role as well. In anaerobic respiration, we're going to concentrate primarily on terminal electron acceptors that contain nitrogen. And this reduction of inorganic nitrogen during anaerobic respiration is going to be really important in the nitrogen cycle. And that's going to be kind of the example we're going to use to illustrate several of these metabolic sequences we're going to talk about, is superimpose them on this phenomenon called the nitrogen cycle, which is critically important for life on Earth. Okay? And we'll show you why that's the case in a second. But you've got to have nitrogen in a usable form. If plants are going to grow, and if plants can't grow, we can't survive. And so we'll use that nitrogen cycle as kind of a unifying theme to talk about some of these metabolic strategies. I talked about that. That's just some stuff about fermentation. So let's get into fermentation just a little bit. Okay? We're familiar primarily with these two methods, or these two processes of fermentation. Alcoholic fermentation or lactic acid fermentation. Alcoholic fermentation takes place in yeast. Lactic acid fermentation takes place in our body, particularly in muscle cells, when we don't get sufficient oxygenation in the tissues to support aerobic respiration. But there are a number of bacteria and other organisms that can also have lactic acid fermentation. What's the difference? What determines whether you can do this or whether you can do this? It's the bottom line. Why can't we go out and run real hard and use alcohol? What do yeast have that we don't have? Right? The right enzyme. Okay. Yeast have different enzymes than we do because they have different genes. Okay. In both cases, however, the crux of fermentation is that we are oxidizing glucose, and we're removing some electrons from that molecule and transferring them to NAD. And as that NAD becomes reduced, the oxidized form of NAD starts to become depleted. It becomes basically a limiting factor in glycolysis. If you run out of oxidized NAD, glycolysis will stop, because that's a coenzyme that some of the enzymes in glycolysis need to have present in order for those enzymes to be active. So the recycling mechanism that works in fermentation just involves an enzyme lopping off some electrons from that reduced NAD and donating them to pyruvate to form, in this particular case, lactic acid. Okay? We have the enzymes to do these particular chemical reactions. But in the case of yeast, they have different enzymes. They have an enzyme that can take pyruvate and decarboxylate it, remove the CO2, to form acetaldehyde. We don't have that enzyme. Okay? And then they can take some electrons from reduced NAD and add those to acetaldehyde to make ethanol. What's our net energy yield from fermentation, remember? 2 ATP net. Remember, you get 4 produced, but there are 2 that were required early on in the steps of glycolysis. So does this recycling step that takes place here generate any additional ATP? No, they were all made during glycolysis. Well, depending upon the organism and the enzyme that it has, pyruvate can have a lot of different fates. For example, there is this genus of bacteria called Propionobacteria. And they have the enzymes that will convert pyruvate into oxaloacetic acid, succinic acid, and then finally, propionic acid. Okay, so this is the end product of fermentation of this particular bacteria. This is the end product that gives cheese, like Swiss cheese, for example. Some of its distinctive flavor is the propionic acid. Okay, so these are used in Swiss cheese making. Clostridium bacteria, which is a different genus, can reduce pyruvate into butyric acid. Anybody know the significance of butyric acid?
I like a really bad smell.
It's a really bad smell. You ever smelled rancid butter? Okay, if the butter is becoming rancid, that's because bacteria are making butyric acid out of the fats in the butter. So metabolizing the fats and making butyric acid. Here we have ethanol being produced by fermentation in yeast. The genus Proteus, which we will use in a number of labs, can convert pyruvate to formic acid, and then also a byproduct of formic acid fermentation is the production of CO2 and hydrogen. So sometimes you produce acids, sometimes you produce alcohols, and sometimes fermentation produces gases as well. It may or may not, depends on the type of fermentation. In these bacteria, Streptococcus, Lactobacillus, Escherichia, sometimes you'll form a number of different types of acids. Okay? If you only produce a single type of acid, like lactic acid, that's typically referred to as homolactic fermentation. If you produce several types of acids, maybe you produce lactic acid, maybe you produce formic acid, then that's called heterolactic fermentation. Sometimes also referred to as mixed acid fermentation. Mixed acid fermenters drastically lower the pH of the environment that the organism is growing in. Now, lactic acid does too, but if you produce all of these mixture of acids, some of which are strong, you can really get a fairly precipitous drop in the pH of your medium as these organisms grow. And there's a test we will do later on in lab called the methyl red test, which is designed specifically to detect this mixed acid type of fermentation. And then another one that we're going to encounter in lab is this one right here. Members of the genus Enterobacter can carry out a type of fermentation pathway that converts pyruvate into acetylmethylcarbinol, and then acetylmethylcarbinol becomes reduced by the NADH, and that's coming from glycolysis, into this compound called 2,3-butadiene, or it's also called acetone. And there's a specific test for this 2,3-butadiene or acetone called the VP test. VP test stands for Vogts-Proskauer test. Actually, in reality, there is a media we'll use called MRVP media. It stands for methyl red Vogts-Proskauer. And we're able to then determine 2 bits of information about our unknown from this one media. Does it carry out mixed acid fermentation? Does it do acetone production? Okay, so those are tests that we will be doing.
Let's get that one for right now.
Fermentation doesn't just have to produce acids and alcohols and gases. It can also produce some organic compounds. Now again, you've got to have the right enzymes to be able to do this, but some species of bacteria can ferment sugars, for example, or nucleic acids, or amino acids, and produce vitamins, steroids, antibiotics. And so medically, these can be some significant compounds. And this is one of the reasons, too, why organisms that can do this are oftentimes beneficial organisms to the host. Some of our common gut bacteria have the ability to produce some of these particular compounds, particularly vitamins. So it's not just a matter of producing just these acids and alcohols. Some other interesting compounds can be produced in fermentation as well. Now, in lab, in a few weeks, we're going to do a number of fermentation tests. And these particular tests involve making a media. You don't have to make it, it'll be provided for you. And that media will contain some broth, and the broth does contain some amino acids. Tryptic soy broth, okay, it's a digestion of protein, and those amino acids can provide both the nitrogen and the carbon and even the energy for the growth of these organisms. If you use amino acids as a food source, one of the things that you've got to do first is you've got to deaminate them. And basically that means chopping off that amino group. Okay. Remember, an amino acid has a central carbon, a hydrogen, a carboxyl group, and an amino group. This R group will vary depending upon what one of the 20 amino acids you are. But in order to use these as energy sources or as carbon sources, you got to take this off first. And when you do that, you produce an organic acid. And organic acids like succinate and fumarate and alpha-ketoglutarate are all part of the Krebs cycle. And so once you've deaminated your amino acid, you can then feed those carbon skeletons into the Krebs cycle and either extract energy from them or maybe convert them into other compounds. What is ammonia going to do to the pH of your test tube? Acid or base? Ammonia is a base, right? So it's going to increase the pH. Now, what we're going to do in these particular tests is, in addition to the broth, we're going to add a sugar, and we want to see if the organisms can ferment the sugar. If they can ferment the sugar, will they still grow in the broth? They should, because they can use the amino acids in the broth. So we're going to give you several different tubes, some of which will contain sucrose that's been added to the broth, or glucose, or lactose, or maltose, or mannose. And we want to see if the organism can ferment these sugars. What if they do ferment these sugars? They're going to produce some kind of byproduct. And more than likely, but not necessarily, some of the end products will be acidic. And that obviously will decrease our pH. Sometimes, when fermentation is occurring, they'll also be producing gases. Those gases could be carbon dioxide, could be hydrogen, but some gaseous compound. Pardon me? In some instances, yes, probably not likely in any of the things we're going to be using in lab. Yep. We'll talk about that strategy, and that's a little different metabolic strategy. Okay, so how can we determine gas production from this particular setup? Well, in the broth tubes, there's a little small inverted tube called a Durham tube. And when you autoclave this media, the autoclaving process drives the liquid into this Durham tube, and so the tube is filled up with broth just like the rest of the tube is. What's going to happen in that Durham tube if you're producing gas during fermentation? Yeah, air bubbles are going to start to displace the broth in that particular tube, and we'll see an air pocket that might develop. Maybe it's just a few small bubbles. Sometimes you displace virtually all the liquid if you produce a lot of gas, and that tube will just kind of float in there. Okay? How are we going to tell whether we're producing something that raises the pH or something that lowers the pH as a result of fermentation? Yeah, okay, we could do a pH test, and we could take a pH meter and stick it in there. But then we'd have to try to sterilize the pH probe every time, not introduce contamination. Built in to the media is a compound that is a pH indicator. And we typically use the pH indicator phenol red. And at a neutral pH, it's kind of red. At a basic pH, it becomes really kind of a dark violet. And at an acidic pH, it turns yellow. So by inoculating these tubes with known organisms and your unknown, you can tell some interesting things. Number one, can it use the sugar as a food resource? Monosaccharide or disaccharide, you know? That's a disaccharide, monosaccharide, disaccharide, disaccharide. disaccharide. In order to use the sugars, you've got to be able to convert the disaccharides into monosaccharides. So you've got to have the enzymes that can break the bond between the two sugars before you can start to ferment them. So if you determine that you can ferment lactose, then you know, hey, my organism has the enzyme lactase. So that's one piece of information. And then you can also say, by looking at the Durham tube and the color of the broth later on, it may or may not produce gases. It may or may not produce acids. Okay. What if you had a tube that you inoculated and it turned nice and yellow in 24 hours, had all kinds of gas in the Durham tube? What would eventually happen to the color of that tube if you stuck it back in the incubator and left it there for several more days? And remember, we're only adding a finite amount of sugar to that tube. But originally it would be yellow, right? So would you be surprised if you came back a few days later on and the color of that tube started to change to a pinkish color, and then eventually a reddish color, maybe even a violet color? No, you would expect that, because they would have used up the sugar, and now they would start switching over and start using the protein, the amino acids in the broth, deaminating that, and if you produce enough ammonia, that's going to raise the pH, and then the pH indicator will turn color. So those are some examples of types of fermentation tests we're going to do. And that's going to be pretty important information for identification of your unknown, because particularly in Berge's Manual 7th edition, there's a lot of reference made to the fact that is able to ferment glucose and produce acids and gas, or is unable to ferment glucose, can ferment lactose or can't ferment lactose. And so that biochemical information is really pretty critical and pretty important. There are some chemical reactions in regard to nitrogen that are important. I've also already talked about one of them. Here are 3 chemical reactions involving nitrogen metabolism that you should be aware of, because we're going to make reference to some of these in the nitrogen cycle in just a little bit. There is amination, transamination, and deamination. Already mentioned deamination. Deamination involves taking amino acid and knocking off the amino group, forming an organic acid, in this case alpha-ketoglutarate, one of the compounds in the Krebs cycle, and then producing ammonia. Amination is just the opposite of that. That involves taking an organic acid like pyruvate or alpha-ketoglutarate or something else in the Krebs cycle and adding ammonia to it to form amino acids. This is one reason why plants are absolutely required to have a source of ammonia to support their metabolism and growth. Because plants, when they fix CO2 during photosynthesis and make sugar, can then easily convert sugar into pyruvic acid, for example, by glycolysis. But how do you make protein? You need some source of nitrogen from the soil in order to be able to then aminate those organic acids to make amino acids. So we're going to talk about the importance of ammonia and the nitrogen cycle supplying ammonia to plant roots to support their growth. Transamination, we actually can do this. Transamination involves taking an amino group from one amino acid and joining it to an organic acid to make a different amino acid. So basically through this metabolic pathway, aspartic acid can be converted into glutamic acid. Now, we don't have the ability to do just any transamination. So for example, if we could convert aspartic acid into glutamic acid, we would not necessarily have to ingest glutamic acid. It would be one of the essential amino acids. But if you didn't have a particular transamination pathway, you might have to ingest that particular amino acid yourself because you can't make it by this particular sequence. Okay, so let's put this together now, some of these things together, and talk a little bit about the nitrogen cycle. Nitrogen has got to be present in a usable form in order for plants to grow. There's just no getting around it. And because plants are the primary producers in the ecosystem, if plants can't grow, we can't survive. Okay? Because we are chemoheterotrophs. We're getting our carbon and our energy from organic molecules. Where do we get those organic molecules? We consume other organisms. Now, if we're a strict carnivore, we consume the tissues of another animal. But where did that organism get its material? Ultimately, it comes back to plants. Okay? The producers have got to make those key organic molecules, and then other organisms can feed on them. Well, nitrogen is a very limiting factor to plant growth. If soil becomes depleted of the wrong forms of nitrogen, plants won't grow. And that's kind of ironic because nitrogen is very, very common in our environment. The air, the atmosphere, is about, they say 79%, I thought 78% of nitrogen gas. But nitrogen gas is notoriously stable. It's one of the noble gases. And plants cannot use nitrogen gas in their metabolic processes. It's like it's not even there. Therefore, nitrogen gas has to be converted into other forms of nitrogen to be able to support plant growth. So we have this very complex worldwide cycle that basically involves nitrogen gas in the atmosphere being converted into other forms of nitrogen, some of which plants can use. But ultimately, if we're going to have a cycle, the nitrogen's got to be returned back to its original form, N2 gas. And so there are several diagrams that you'll see in the next few slides which are representations of the nitrogen cycle. And one of the things that you'll see is that the nitrogen cycle involves a whole bunch of different types of microorganisms. And what's driving the nitrogen cycle is metabolism. These organisms are using nitrogen for sources of energy, producing byproducts, and some of those byproducts will be useful for other organisms' metabolism, including plants. There are four basic types of reactions that make up the nitrogen cycle. They're nitrogen fixation, ammonification, denitrification, and nitrification. And typically, one or a group of organisms only does one of these things. So it's not like we have one bacteria that's a nitrogen fixer, also an ammonifier, a nitrifier, a denitrifier. Typically, the nitrogen cycle involves this complex interaction between all kinds of different organisms. And the waste product of the metabolism of one organism is the food, if you like, of another organism. So, nitrogen fixation, what's that called? That's a process whereby this atmospheric, this stable N2 gas is converted into ammonium. And typically it's ammonium salts. The enzyme that does this is the enzyme nitrate reductase. And so it grabs hydrogen basically and donates it to N2 gas, reducing it into ammonia. Now, not all species of bacteria have the enzyme nitrate reductase. The enzyme itself is highly oxygen sensitive. The enzyme basically is poisoned by oxygen. Back to that in just a second. There are two general categories.
Is nitrogenase and nitrate reductase are the same thing?
Oh, I'm sorry. I used the wrong enzyme. Thanks for catching that. Nitrogenase is the enzyme. Nitrate reductase is a different one. Thanks for keeping me honest there. There are two types of nitrogen fixation, two categories. There's symbiotic nitrogen fixation and non-symbiotic nitrogen fixation. The non-symbiotic is carried out by three living microorganisms. Several of them are cyanobacteria. Remember when we talked about cyanobacteria, we said that some of the filamentous cyanobacteria had the specialized cell, the heterocyst, in the filament? That's the environment in which this nitrogen fixation takes place, because there are adaptations in the heterocyst that create basically an anaerobic environment within that cell. And so it's an oxygen-free environment. Symbiotic nitrogen fixation primarily takes place within the roots of the family of compounds legumes. So clover, peas, alfalfa, beans. And within the roots, nodules develop. They look like little tumors almost on the roots. And that's where the bacteria live. And within the nodules, there's a bunch of the compound leg hemoglobin. Leg refers to legging. What's the purpose of leg hemoglobin? Okay, remember that this enzyme, this nitrogenous enzyme, is oxygen sensitive. What's that like hemoglobin going to be doing? It's going to be binding up to free oxygen, okay? And therefore, this contributes to kind of an anaerobic environment in those root nodules that allow these anaerobic bacteria to carry out this process of nitrogen fixation. This particular symbiotic relationship is a type of relationship called mutualism. Mutualism is a win-win relationship for the organisms involved. If we use pluses as being benefit, both organisms in the partnership benefit. Obviously, what do the plants get? They get the ammonia that they need to be able to metabolize and produce proteins and nucleic acids. What do the bacteria get? What do they get in those while they're in those plant roots? Sugar. Okay, plants are photosynthesizing. So this is a critical process that must take place. Now, there are some strictly physical processes that can actually cause the reduction of nitrogen gas to ammonia. And one of them is very high energy discharge. A small amount of ammonia can be produced during lightning storms. And then when it rains, that ammonia will wash into the soil. By far, the most significant amounts comes from this either symbiotic or non-symbiotic nitrogen fixation. Okay, ammonification. In ammonification, what happens is bacteria and other decomposers are breaking down amino acids and deaminating them. And that's part of their metabolic strategy, is they're then using those organic acids as food sources. The ammonia that is produced can then leach into the soil. You ever gotten a good sniff of some ripe roadkill? Or come across a dead animal in the woods? What do? You get that real whiff of ammonia. That's the result of the deamination of the proteins, the muscle tissue, etc., in that decomposing animal, and those ammonifying bacteria chopping off that amino group and then using the organic acids as energy sources. Nitrification involves the conversion of ammonia into either nitrate or nitrite. And that's carried out by a group of organisms called the lithotrophs. So we're going to have to backtrack just a little bit before we continue and talk about this particular strategy of lithotrophy. Another name for that particular strategy is chemoautotrophy. So lithotrophs and chemoautotrophs are the same type of organism. These are really fascinating organisms. These are organisms that defy the normal logic that all life depends directly or indirectly on the sun. These organisms do because they're phototrophs. These organisms do because they're phototrophs. These organisms do because they're using organic molecules of their source of carbon and energy, and they came from ultimately photosynthesis. But these organisms do not. These organisms are using CO2 as their source of carbon. CO2 organic or inorganic? It's inorganic, right? These organisms are using compounds like nitrate as their source of energy. They're pulling electrons off of nitrate, for example, or off of ammonia, or off of nitrite. Organic or inorganic? Inorganic. So these are organisms that subsist entirely on inorganic molecules. If the sun were to burn out, everything on this planet would die except these lithotrophs, these chemoautotrophs, because they could use the CO2 from the atmosphere, and they could literally decompose material in the rock, these minerals, as their source of energy. Lithotrophs are also called the rock eaters. So these are exceptions to the normal rule we always kind of spout to you. So we'll dig into, just as an aside, lithotrophs just a little bit, and then come back and finish the nitrogen cycle. So don't forget to pull plates out, just stick them in the fridge, and then get your unknown going into broth sometime.
I have two questions. In the comparison between aerobic and anaerobic respiration during the electron transfer chain, on that diagram it showed that the first or the start of the electron transfer chain was different. Like the arrow was pointing to a different part of the chain.
Let me look at that diagram and see, because I shouldn't be.
Yeah, because that was actually, it might be on the side notes. It was right here. So this is pointing here, and this one's pointing right there.
Oh,
and I was wondering what would cause the difference between those two, because nothing else in the chain up to that point.
You know, I'm not sure that the author of that really was trying to indicate. I think they were careless with the placement of their arrow, really, because reduced NAD will jump into the electron transport chain at a different point than reduced FAD.
Okay.
But in terms of there being a fundamental difference about where electrons come from NAD or FAD in aerobic or anaerobic, there isn't a difference, at least to my knowledge.
Okay.
So I think this is an example of the artist being a little sloppy and not understanding what they were drawing.
I figured it was a diagram issue, but
Yeah, it is a diagram issue, yeah.
Just in case, it was like, where would the disparity come in?
Right.
Because
yes,
how would this?
Absolutely.
How would it know? The other question is if, and this is kind of an out there question, but
okay, good.
If there was